Okay, good morning. It's time to start with this plenary session on druggable mutations and other personalized approaches for the treatment of MDS. I will call the first speaker, who is, hello, Stein from New York, who will discuss IDH1 and 2 mutated MDS from gene to therapy. By the time Ethan gets to the mic, I would just like to let you know that for the dinner tonight, there are easy public transportation to go to the restaurant, so there is no issues with that, and you are very welcome for this dinner in a very nice and pleasant place. So, Ethan.
Okay, thank you so much for the kind introduction. Thank you to the organizers for inviting me to speak at this wonderful meeting. It's not that often that someone who specializes in AML gets invited to MDS meetings. I feel honored that I'm allowed to be here, so thanks again. So what we're going to be talking about today are IDH1 and IDH2 mutated myelodysplastic syndromes with a particular focus on the role of IDH inhibitors when you encounter a patient with myelodysplastic syndrome that has an IDH mutation. Here are my disclosures. So the four topics I wanted to talk about today, or three topics, are as follows. Number one, how common are IDH mutations in patients with myelodysplastic syndromes? Is there a role for IDH inhibitors in the treatment of newly diagnosed and relapsed and refractory IDH mutant myelodysplastic syndromes? What is the role of IDH inhibitors for patients with IDH mutant clonal hematopoiesis with cytopenias, otherwise known as Sekeres? And we're going to focus mainly on IDH2, because although there is data in abstract form for IDH1, there is actually published data for IDH2 mutations. So one thing to highlight is that IDH mutations are actually pretty uncommon in patients with myelodysplastic syndromes. If you compare the amount of the frequency of mutations in AML as compared to MDS, you can see it's about threefold lower. So in AML, about 15% of patients will have an IDH2 mutation, and about 7.5% of patients will have an IDH1 mutation. You can see that for MDS, it's about 5% in both categories. That's not only for myelodysplastic syndromes, but also for myeloproliferative neoplasms. So how do IDH mutations lead to the development of myelodysplastic syndrome? So we think about it in about the same way we think about it in patients with acute myeloid leukemia, that when you have IDH1, which is found in the cytoplasm, or IDH2, which is found in the mitochondria, that mutant enzyme leads to the production of beta-hydroxyglutarate. If you can then lower the levels of intracellular beta-hydroxyglutarate, you then get the ability of those cells to differentiate into normal, healthy neutrophils. Now, I always show this slide in every talk that I give, but I usually show it in the context of acute myeloid leukemia. And one thing that I think we've all observed in patients who've had a prior myelodysplastic syndrome, who progressed to acute myeloid leukemia with an IDH mutation, and then you give them an IDH inhibitor, that IDH inhibitor is very, very good at reverting the patient back to their underlying myelodysplastic syndrome. That is, it's very good at leading to marrow CRs, but it doesn't necessarily or always lead to hematologic improvement. So a common clinical manifestation of what we see when we have these patients is that you'll give them an IDH inhibitor, they'll have some brief improvement in their blood counts and lowering of their blasts to less than 5%, but then they will become transfusion dependent once again, because essentially what you've done is you've revealed the underlying MDS and stripped away the overlying acute myeloid leukemia. So that's a very long way of saying that in order to cure IDH mutant myelodysplastic syndromes, IDH inhibitors as single agents are probably not going to be the answer. So what is the data for using IDH inhibitors in patients with MDS? So again, focusing just on IDH2 inhibitors, this was a clinical trial that actually was a very small subset of the large phase 1, phase 2 clinical trial that led to the approval of enasidenib for IDH2 mutated relapsed and refractory AML. In that trial, if you look at the top, 239 patients were enrolled, and of those 239 patients, 17 patients had MDS. 10 of those patients were enrolled in the dose escalation portion of the study. 7 patients were enrolled in the dose expansion part of the study. I want you to remember that because I think it's going to help explain some differences you're going to see in the response rates of earlier trials that were done earlier for IDH2 mutant MDS and trials that were done a little bit later. Here are the baseline demographics of the 17 patients that were treated on the trial. You can see their median age is 67 years old. If you look at their IPSSR classification, most of them actually have very low or low-risk disease or high or very high-risk disease. And then when you look on the left-hand side at their responses, there are a number of things which are informative. The first, or the middle column, is looking at all patients that were treated, those 17 patients. And then the right-hand side are the patients who had previously been exposed to a hypomethylating agent. So when you look at the overall response rate, you can see that it's somewhat admirable at 53% in all patients treated and 46% in patients who had a prior hypomethylating agent. But is it really that admirable? I'm not so sure. When you look at the rate of complete remission, the strict definition of complete remission, which I think is to some extent what's important in MDS, or certainly getting some hematologic improvement, none of the patients achieved a true complete remission. There were a few patients who achieved a marrow complete remission with hematologic improvement. I think that's reflected in the swimmer's plot on the right-hand side, where you can see of the first 4 lines, these were patients who had either a marrow CR or either a hematologic improvement or a complete remission. You can see generally these are the patients who were able to remain on study the longest. It's also notable to point out that even those patients who were able to remain on study, they eventually, within the course of 2.5 years, ended up developing progressive disease. Again, arguing to the point that a single agent IDH inhibitor may be an initial strategy to treat these patients, but it's not going to be the overall answer. Now, Dr. DiNardo and her colleagues at the MD Anderson Cancer Center did a larger clinical trial looking at the use of enasidenib in 2 different clinical settings. One clinical setting, which was Arm B, were patients who had previously received a hypomethylating agent with an IDH2 mutation, had progressed or had not responded, and then went on single agent IDH2 inhibitor enasidenib. And that's what I'm showing you in this slide. You can see on the left-hand side, the median age of these patients was 73 years old. This patient population included some patients that we would now call acute myeloid leukemia, 2 patients, so we'll remove those patients for a second, and 3 patients with chronic myelomonocytic leukemia. But if you look at the rest, there are then 18 patients with relapsed or refractory IDH2 mutant myelodysplastic syndromes. On the right-hand side, I think you can see something that's informative and different than what I showed you on the prior slide. And that is that 5 of these patients achieved a stringent MDS working group criteria complete remission. So why is it that patients are achieving a complete remission on this study? And in the prior study that I showed you, there were no patients achieved a complete remission. So I would argue this has nothing to do with science, and this has to do with the cultural characteristics of enrolling patients and treating patients on clinical trials. When that original trial that I showed you a slide ago was done, we really didn't know. In retrospect, I don't know why we didn't know how to use IDH inhibitors, but at the time, we were doing things like pulling patients off study relatively quickly. If they didn't have a quick response, we said, it's not working, we're going to take them off study. And those patients probably didn't have the opportunity to achieve a complete remission. I think by the time Dr. DiNardo and her colleagues did this study and then published it, I think we understood that as a differentiation agent, patients needed to stay on an IDH2 inhibitor for a longer period of time. And I think that's why the response rates here are significant. I mean, they're better than what I showed you in the previous slide. Dr. DiNardo and colleagues also had a separate arm of the trial where she enrolled 27 patients with newly diagnosed IDH2 mutant myelodysplastic syndromes and gave them the combination of enasidenib with azacitidine. So now I want you to hearken back to an AML trial that was done. Some of you might not read AML trials, but you should because they're interesting. So an AML trial that was done a number of years ago, a randomized trial where patients with IDH2 mutant acute myeloid leukemia were randomized to receive either azacitidine alone or azacitidine with enasidenib. And the top-line results of that trial is that there was no benefit to adding enasidenib onto azacitidine. There was no overall survival benefit. There was no event-free survival benefit. There are many caveats, and there are reasons we can say that it didn't show a benefit, but those are the top-line results. So the question here was, what happens if you add enasidenib to azacitidine for myelodysplastic syndromes? Again, on the right-hand side, what you can see in the 27 patients is that the overall response rate is higher at 74% than what I showed you in the relapse refractory setting. The complete response rate or the complete remission rate is similar, though. It's 26%. And really what you're getting, just if we go back for a second, so you can see in the relapse refractory patients, there were no marrow CRs, but in this patient population where you are giving enasidenib in combination with azacitidine for newly diagnosed patients, the rate of marrow CR goes up, so 9 patients achieve a marrow CR, and I think that's what's driving the improved overall response rate. I also think it highlights what I told you before, which is that this drug, enasidenib, is very good, I think, at clearing out the blasts of anyone, whether you have acute myeloid leukemia with an IDH2 mutation or myelodysplastic syndrome with an IDH2 mutation. I think it's less clear whether it improves cytopenias for the long haul. The other thing this trial doesn't answer, which I think is something that needs to be answered, is what does adding enasidenib to azacitidine actually do? That is, if you just gave single agent enasidenib, could you get results that are similar to what you see when you combine enasidenib with azacitidine? When you look at the molecular features of the patients that were treated on this trial, I just want to point out a couple of things. So one is that the molecular features of the patients on this trial, so these are newly diagnosed patients and relapse refractory patients, but the molecular features are very similar to the molecular features that you see in patients with relapsed and refractory acute myeloid leukemia. That is, you have a preponderance of patients who have additional mutations in splicing factor genes. And it raises the possibility that when you're seeing these splicing factor gene mutations, which may be the main driver of the myelodysplastic syndrome, that in order to really change the natural history of this disease, you're going to need to do something where you're giving a therapy that addresses the splicing factor mutant component of the disease in order to get durable complete remissions. Okay, I'm just 1 second on IDH1 mutant myelodysplastic syndromes. Again, there isn't a lot of published data. There are clinical studies in Europe and the United States that are investigating IDH1 inhibitors in patients with myelodysplastic syndrome. There's a small amount of published data on the use of a new IDH1 inhibitor, olutasidenib, in patients with MDS. They kind of snuck it in to their Lancet Hematology paper for relapsed and refractory AML. There are ongoing studies in the United States, one sponsored by Servier, and in Europe, the IDIOM trial, which has been presented a couple of times. And there are preliminary results that have been presented in abstract form, have shown activity similar to IDH2 inhibitors in IDH2 mutant patients. Okay, for the last 2 minutes, I want to talk about the role of using IDH inhibitors to prevent overt myeloid malignancies. Unfortunately, I only got here yesterday, so I don't know if anyone talked about this already, but I'm sure someone did. But if they didn't, you should know that when you have an IDH mutation in the setting of clonal hematopoiesis, the chances of going on to develop an overt myeloid malignancy are quite high. That was shown in a number of trials, one published by Mikkael Desai in Nature Medicine, which I'm depicting here, the other that was published by Sagi Abelson and Liran Shlush in Nature around the same time. We have designed a clinical trial giving enasidenib, the IDH2 inhibitor, or ivosidenib, the IDH1 inhibitor, for patients with clonal hematopoiesis with cytopenias. This is being done in conjunction with Dr. Kelly Bolton at Washington University. She's doing the IDH1 component. I'm doing the IDH2 component. We're giving 15 IDH2 mutant CCUS patients enasidenib and then monitoring for their response to therapy. In addition, Kelly's going to be doing some correlative work looking at the clonal architecture and single cell mutation analysis of the patients who are treated on this study, and then also doing some mouse work with a mouse model of the same entity. Since this is not being sponsored by a pharmaceutical company and I'm not beholden to any investors, I'll tell you what's happening on the study. We've enrolled 2 patients on the study so far, 2 out of the 15 with IDH2 mutations. Both of the patients have had a remarkable hematologic response to treatment. Okay, so their counts have all gone up dramatically, and they've normalized their blood counts. In addition, the variant allele, they both also have SRSF2 mutations, so they're IDH2, SRSF2 co-mutated. Their blood counts, as I said, have normalized. The variant allele fraction of IDH2 has been decreasing steadily, so it decreased steadily through the first 6 months of treatment, and now that's all the data we have right now. And I think most importantly, and something that everyone has been worried about, is that will you see new mutations pop up because of the selective pressure of targeting IDH2? And am I going to make their disease worse by now causing them to have clonal hematopoiesis or CCUS that is primarily driven by a splicing factor mutation? And the answer is we've not seen any new mutations pop up. We've not seen a rise in the variant allele fraction of the other mutations either. The last thing I'm going to tell you is something that Dr. Bolton, Kelly Bolton, has done. I don't know if she's in the audience, but the Kelly Bolton has done, which I think is really, really impressive, and we're going to be doing it for the IDH2 inhibitor trial as well for CCUS. So Kelly had this idea that in the age of COVID, where everything is virtual, and when you have a mutation that is really so rare, the only way to do a trial like this is by doing it virtually. You can't expect people to fly into New York and to fly into St. Louis to enroll on the trial. Because I am not very forward-thinking, told Kelly that I thought she was crazy, it would never happen. However, she is persistent, and she really persisted, and she has made this fully virtual trial happen. So what happens is that patients actually enroll on the trial wherever they might live in the United States. Their local oncologist does all of the standard of care testing, because that's really all that's being done for these patients, except for correlative studies that are being collected by a mobile Quest van, not one van, but multiple vans, that go to the patients' houses, draw their blood, and then send those samples to Washington University in St. Louis. The study visits are done remotely with someone, either me or sitting in New York, or Kelly sitting in Washington, or not in St. Louis, excuse me, at Washington University in St. Louis. And I think this is really going to allow the enrollment of these patients relatively quickly. We're thinking of ways we can expand this internationally. We've already had some inquiries from Saudi Arabia and other Middle Eastern countries, and we're very, very excited about this program. So in sum, I've gone over by 2 minutes and 28 seconds. IDH inhibitors have a role in the treatment of MDS, both as single agents and in combination with azacitidine. I think the benefit of AZA in combination with IDH inhibitors over IDH inhibitors alone has yet to be proven in a large study. The molecular landscape of IDH2 mutant MDS is similar to IDH2 mutant relapsed and refractory AML. IDH2 mutations often co-occur with splicing factor mutations. There's a rationale for dual targeting. Of course, we need something that addresses splicing factor mutations before we can dually target anything. And I think the next frontier, and the one I'm the most excited about, is the primary prevention of overt myeloid malignancies by targeting IDH mutant clonal hematopoiesis and CCUS. And with that, I thank you very much for your attention and look forward to your questions at the end of the session.
Thank you very much. I will ask the next speakers to stick to the time because there is another session just after this one. And if you want to have time for the discussion, it will be important to stick to the time. So the next speaker is Mikkael Sekeres from Miami, and he will discuss the therapies for MDS with Del5Q. Thank you, Mikkael.
Merci beaucoup. I want to thank the organizers for inviting me. It's really a privilege to be here. And now that you said that, you've made me really nervous, so I'm going to speak at twice the pace I usually do and finish in 7 minutes. I'm going to talk about current treatment of MDS with deletion 5q. Now, do I get extra time because the clicker's not working? All right. I'll go over briefly classification and risk, treatment of lower risk, and then higher risk MDS with deletion 5q. So we will have a spirited discussion tomorrow about the latest iteration of the WHO classification and the ICC. If you're using the WHO classification and looking for deletion 5q, I put a big fat red arrow where it's located. For lower risk patients with fewer than 5% bone marrow blasts, alone or with one additional abnormality except for -7 or deletion 7q. You could also, I didn't do that. Is this a psychological experiment to see how often I press the clicker to move it forward? Is it working? Oh, look at that. It's moving up there, but not on my screen. I think I'll take this stance. Interesting. Okay, so if you're looking at I get like an extra minute for all this, by the way, right? So if you're looking at the WHO and thinking about higher risk MDS, you can also have deletion 5q, and it would fall somewhere in the area of where that other big fat red arrow is. For the ICC, my monitor caught up to me, you would also have a group called out with deletion 5q, with up to one additional abnormality, again accepting chromosome 7 abnormalities, and this time also accepting multi-hit TP53 abnormalities. And again, if this were higher risk disease, it would fall somewhere in the area of that second red arrow. Now, having additional TP53 is meaningful. This was a study published about a decade ago looking at 55 patients with deletion 5q abnormality, 10 of whom had a TP53. The median overall survival for those without and with a TP53 mutation were 73 versus 57 months. TP53 mutations with deletion 5q are bad. But they're not the only mutation that you can see with deletion 5q. This is a study we published about 5 years ago looking at 178 patients with deletion 5q, 43 of whom had an isolated deletion 5q, 46 with excess blasts. You can see that TP53 seemed to be represented out of proportion in patients with deletion 5q compared to those without deletion 5q. You could see other mutations, including DNMT3A, TET2, SF3B1, etc., but in lower proportions than you saw in patients without deletion 5q. And of course, those with the TP53 mutation had a worse overall survival and were more likely to have higher risk MDS. Let's talk about the treatment for patients with lower risk MDS. This is one treatment algorithm, something that I published with Bhumika Patel in the ASH Education book in 2019, that divides treatment of patients with lower risk MDS into different categories depending on their predominating cytopenia at the time. If you go down the branch that says anemia, you go through erythropoiesis-stimulating agents or luspatercept and land on lenalidomide for those with deletion 5q. But it's just as reasonable to use erythropoiesis-stimulating agents in these patients. On the left is a meta-analysis that we published, gee whiz, about 15 years ago, looking at 20 years of published literature of ESAs in MDS, specifically for lower risk MDS, reporting an overall response rate of about 40%. On the right is the randomized trial conducted in Europe, led by Dr. Plotzbecker, in which patients received darbepoetin or placebo. 11% of these patients had deletion 5q. Looking at a longer period of time of observation, the hematologic improvement rate was 35%. What about luspatercept? This, of course, is one slide looking at the transfusion independence response rates for patients who were treated with luspatercept who had ring sideroblasts. Those were 38% of those who received luspatercept versus 13% in the placebo group. But if you go to the clinicaltrials.gov website, you'll see that patients with deletion 5q were excluded in this study. Now, there are a couple of reasons that I can think of that patients with deletion 5q are excluded from these studies. One may be that there is truly some biological variability in patients who have deletion 5q versus other types of MDS. Another reason may be that a single manufacturer makes a variety of drugs, including one for patients with deletion 5q, and don't want to compete with their market share for patients who receive that drug with deletion 5q and therefore exclude them from these trials. I think this is a real problem in how we're doing drug development in lower risk MDS, and we should probably insist on patients with deletion 5q being included in these studies unless there is a true biologic reason they shouldn't be. Boy, I had to dust off some old slides for this presentation, and this is one of them. This is a collection of patients from the MDS 001, 2, 3, and a PK study that was conducted focusing on those patients who had deletion 5q. This was presented by Alan List at ASH in 2006. You'll see here that 74% of patients who went on to these studies with lenalidomide had been exposed previously to erythropoiesis-stimulating agents. So it's just emphasizing the point that just because a patient has a deletion 5q with anemia, we don't have to spring right to lenalidomide to treat them. We can actually start with an erythropoiesis-stimulating agent. Across these studies, 76% of patients had an isolated deletion 5q, 16% had an additional abnormality, and a paucity of patients had complex karyotype because enrollment criteria tended to be lower risk MDS as defined by the IPSS. Erythroid response rates were 68% for transfusion independence with treatment of lenalidomide. This was approximately double what was seen for any intervention for lower risk MDS at the time, and that's why we all got excited about it. This is, again, the division by IPSS score of patients across these studies. The majority of patients had lower intermediate 1 risk MDS, and the majority of patients had an isolated deletion 5q plus 1 abnormality with a paucity of patients having complex karyotype. I show these slides to say when you're looking at transfusion independence response, it's probably better in patients who have an isolated deletion 5q. It's still good in those who have 5q plus 1 abnormality. It's probably not very good in patients who have complex karyotype, but not a lot of these patients were enrolled to these studies. What predicts response? This is a publication we had about 15 years ago showing that those patients who did not have baseline thrombocytopenia but did develop thrombocytopenia with a drop in platelet count of over 50% were highly more likely to have an erythroid response to lenalidomide when they had deletion 5q than those who didn't develop that amount of profound thrombocytopenia. The same was true in patients who did have baseline thrombocytopenia before they started treatment with lenalidomide. Results were a little more mixed for neutropenia. It appeared that those patients who did not have baseline neutropenia but had a drop of over 75% of their neutrophil count were more likely to have an erythroid response to lenalidomide than those who didn't. This is kind of speaking to more of an effect that is truly disease modifying and an effect on the clone that then leads to the improvement in erythroid counts. In multivariable analyses, again, the decline in platelet count came out, as did younger age, shorter duration of MDS prior to study entry, and lower baseline transfusion burden. Allen did a similar multivariate analysis and found that lower risk IPSS, less karyotypic complexity, and having the 5q minus syndrome were also associated with an erythroid response to lenalidomide. Overall survival for patients is better when treated with lenalidomide when they have an isolated deletion 5q versus more complicated karyotype. And these are bottom line results or top line results for the randomized trial in which patients treated with lenalidomide at higher doses had the best transfusion independence rate at 61% compared to 6% for placebo. Median duration of response is over 2 years for deletion 5q treated with lenalidomide. And we did an interesting study where we combined data from the phase 2 trial and the randomized trial and looked at dosing and what dosing actually predicted not only response but overall survival. Prediction of overall survival. I thought this was really interesting. And it basically boils down to the philosophy that you give an induction dose of lenalidomide during the first 3 cycles, where you give dose lenalidomide at high doses, 10 milligrams, 21 out of 28 days or 28 out of 28 days, and then you dose reduce based on cytopenia after those first 3 cycles. So think of it as an induction course and then a post-remission course. When you follow that strategy, patients actually live longer. So going back to the data with TP53, Remember that 10 patients in this cohort of 55 patients had a TP53 abnormality. 37 were treated with lenalidomide, and 8 of the patients with TP53 were treated with lenalidomide. TP53 treatment with lenalidomide had no karyotypic response and a trend to a higher rate of AML progression. So in patients who have lenalidomide with a TP53 abnormality, lenalidomide is a lot less useful than in patients without the TP53 abnormality who had deletion 5q. Okay, so there were some results presented recently looking at treatment of lenalidomide before patients actually become transfusion dependent. And I think this kind of echoes what we just heard from Eitan about thinking about starting treatment earlier in a patient's disease course. In this study, patients had to have lower risk MDS without red cell transfusion requirements, and they had to have a deletion 5q abnormality. They were then randomized to receive lenalidomide at 5 milligrams per day for days 1 through 28 or placebo. When we follow these patients over time, there was a significant reduction in the, significant extension in the time to transfusion dependency among those patients who were randomized to lenalidomide versus those who were randomized to placebo. And you can see that the median hemoglobin, of course, was better in those who were treated with lenalidomide, and treatment with lenalidomide was associated with a karyotypic response. Now, there wasn't a difference in median overall survival, interestingly, in these patients. And of course, one of the great concerns we have is that the earlier you expose a patient to lenalidomide, the more likely they are to evolve resistant mutations like TP53. So there were patients who evolved TP53 mutations in the group treated with lenalidomide. There was also a patient who evolved in the placebo group. So my take on all of these data, I am not yet ready to start lenalidomide in patients who aren't yet transfusion dependent. I think what we're really looking at is a philosophy of treating patients with lenalidomide upfront to avoid that very first transfusion. Because once our patients start to become transfusion dependent, we tend to start lenalidomide. I don't think in personal philosophy that it's worth it to a patient to expose them to this drug for weeks or months or even longer to avoid that very first blood transfusion. But others may have a different philosophy about this. Until I see improvement in overall survival data or transformation to AML data, I think I'll hold off in doing this. Now, what about use of hypomethylating agents in patients? This is from the study we conducted looking at attenuated doses of azacitidine and decitabine, so 3-day dosing of each of these in a 28-day cycle that was in NEJM evidence. In this study, only 2% of patients had the deletion 5q abnormality. So it is something I will turn to in patients if they have an isolated anemia after I've tried ESAs and lenalidomide. For patients with multiple cytopenias, I might turn to it a lot earlier if I want to, if I have cytopenias that I really need to deal with. Okay, so moving on to higher risk MDS with deletion 5q. This is again a typical treatment algorithm for patients with higher risk MDS, where it usually has 2 branch points, funneling patients towards being treated with hypomethylating agents or towards transplant. Of course, this is the AZA-001 results showing a survival advantage for patients with higher risk MDS treated with azacitidine versus treated with conventional care regimens. I do think that that is the standard of care for patients who have higher risk MDS with a deletion 5q abnormality. But you may ask the question, what about treating these patients with lenalidomide also? Well, the GFM, in this case led by Dr. Ades, tried to answer this question, looking at patients with deletion 5q with higher risk MDS who were treated with lenalidomide. Of the 47 patients in this study, 29 had MDS. The CR rate was 21%, all in patients with an isolated deletion 5q abnormality, not in those who had additional karyotypic abnormalities. Among 18 AML patients, there was only 1 CR. And here's the survival curve, which is frankly kind of depressing. I mean, the survival is less than a year. We looked at this also in the Southwest Oncology group, giving high-dose lenalidomide at 50 milligrams per day to patients with AML who had the deletion 5q abnormality. Our overall response rate was 14%, and here you can see that there were responses in patients who had a complex karyotype. What about combining lenalidomide and azacitidine? Well, we thought that this would really do wonders for patients who had a deletion 5q MDS with higher risk MDS, and you can see here is the overall response rate, 62% for those who got aza and lenalidomide versus 50% for those who got azacitidine monotherapy, so not a statistically significant difference of adding lenalidomide to azacitidine. So what can we conclude from all these data? Well, deletion 5q MDS can be heterogeneous, and it's not all good risk. For low-risk deletion 5q MDS, treatment includes ESAs, lenalidomide, attenuated dose hypomethylating agents, and maybe others. For higher risk deletion 5q MDS, HMA monotherapy and transplant are still the best treatment options for now. So I want to thank all of you for listening today, and our fabulous team at Sylvester Cancer Center, University of Miami, and of course, always our patients.
Thank you very much, and thanks to you, we are now on schedule. Almost. So it's a pleasure to announce the next speaker, who is Jeffrey Tyner, who will discuss the use of functional genomics for the development of new therapeutic approaches in AML and MDS.
Well, thank you very much to the organizers for the opportunity to present at this outstanding conference. I do have some disclosures. I've received, collaborated with and received research funding from a number of pharma and biotech companies, and I'm on a scientific advisory board for Recludix. So I'd like to tell you in the next 15 minutes about some of the work that my team has done at Oregon Health Science University in Portland, Oregon, to use an integrative approach towards studying myeloid malignancies. And we've primarily used this approach to study AML, but I'm going to try, in the context of telling you about our AML work today, to highlight a few of the data points that are most interesting and relevant for the subset of our cohort that has secondary AML arising from MDS. This integrative approach has attempted to study these tumors holistically, such that we can understand both the tumor intrinsic genetics, signaling pathways, processes, and also how those are cross-talking with the tumor microenvironment, such that we might arrive at improved drug combinations for therapy. This approach has relied on a variety of functional platforms, some of which we developed in-house, others we incorporated work from others. This involved genetic and pharmacologic perturbations of primary specimens, blood and bone marrow specimens from AML patients, with a variety of different readouts to understand how these perturbations are impacting on the tumor cells. Much of what I'll talk about today, most of it will be high-throughput readouts that assess sort of the aggregate bulk viability of the tumor. And I think everyone here will understand that this is the main reason we've applied this to AML. It would not work very well for a primary MDS specimen. However, we are evolving, doing away somewhat with the high-throughput approach, moving towards imaging-based, flow cytometry-based, single-cell sequencing-based assays, and I'm very excited about the forward-looking opportunity for those new readouts that would allow us to actually implement some of these functional platforms for primary MDS specimens. We can couple these functional perturbations with genomics, whole exome, RNA sequencing, et cetera, and through a lot of computational work, primarily by my fantastic colleague, Dr. Shannon McQueenie and her team, we can integrate all of these data, model pathways, and understand mechanisms and biomarkers of drug response and drug resistance. So I'm going to talk largely about our Beat AML program, the 1.0 version of this program, which is a preclinical effort where we endeavored to collect a large cohort of AML patient specimens, perform these functional genomic assays, computationally integrate all of the data to understand, again, markers and mechanisms of drug response that can then hopefully be parlayed into clinical trials. The bulk of these data, the first two-thirds of the data set, were published in 2018 in the Nature paper referenced at the bottom of the slide. And this paper really laid out the functional genomic landscape of this cohort, where we showed all of the sequencing data, all of the ex vivo drug response data, as well as detailed clinical annotations. And from this, we could integrate and understand patterns of mutations that correlate with sensitivity or resistance to drugs, gene expression profiles that could predict drug response or resistance. We could look at all of the gene expression events across the whole RNA-seq data set using an algorithm called WGCNA, categorize these into different modules of gene expression that could be looked at continuously with drug response to extract additional signal. And we could also look at drug response versus clinical features, such as secondary AML versus de novo AML, and see some very interesting differences, and I'll come back to this point a little bit later in the talk. By and large, though, this study established many predictions about biomarkers of drug activity. Some were proof of principle, but many were unknown, and all required mechanistic and clinical validation. And so we've spent a lot of time doing this, and I should say that one of the foundational principles of this program was to democratize and share all of the data, which we've done in many different ways. All of the raw data can be downloaded through the NIH GDC platform. Processed versions are available on GitHub links, and Shannon McQueenie and her team did a fantastic job setting up a point-and-click tool called Visome that one can use to visualize and explore the data set. And so I think largely because the data was shared so broadly, both through internal collaborators at OHSU as well as external individuals leveraging the data set, I can say that the project has had relatively broad impact. Again, the original inception was to understand what are the markers and mechanisms of drug response. We've done a lot of that and just shown her a few examples. We've looked at BCL2 inhibitors, bromodomain inhibitors, variety of kinase inhibitors, et cetera, and been able to show markers as to why these drugs may or may not be working across this patient cohort. There's been a lot of other instances, though, where this data set has been used in ways that we hadn't even conceived of when we first started the program. We didn't expect to find novel recurrent oncogenes, but there are some in the data set. I'm just highlighting one that we've worked up that we'll hopefully publish soon called ZNF687, a transcription factor where we found some recurrent frameshift mutations that are change of function of the gene. We drew inspiration from a lot of the work in the first session this morning, and thanks very much to Dr. Freeman for calling out this study from our group in her outstanding presentation earlier. Anupriya Agarwal, Shannon McQueenie, and investigators at the Knight Diagnostic Labs, although we had no intention of studying germline predisposition, realized that we did have a large data set of germline sequencing, and so we're able to look into that and show that about 10 to 15% of the subjects in this cohort harbor known, likely, or likely pathogenic germline variants. Evan Lind and more recently Jenny Saltz at our institution have used these data as well as some of their own data to dig into the immune landscape of these diseases and shown that there's broad immune suppression, sometimes can be restored with drugs such as immune checkpoint inhibitors. Others have looked into this data set and surveyed the inflammatory microenvironment and shown that there are a variety of factors in this microenvironment that are probably driving disease pathogenesis and modulating drug response. A number of individuals have used these data to either develop or validate new outcome determinants for AML. Chris Oakes at Ohio State has done a terrific job of leveraging about over 200 of these specimens in which he conducted DNA methylation studies and found about 14 different epitypes, DNA epitypes, methylation epitypes in AML that correlate with mutations, correlate with cell state, and with clinical outcome. And a number of individuals have used these data to model drug resistance. So this was the initial tranche of data, the first two-thirds. One of our pandemic projects was to organize and finalize and lock down the final third of the data set and harmonize it with that initial Nature paper. And so we have done that and published in Cancer Cell last August. The original two-thirds in the Nature paper is what we call waves 1 plus 2. The final third of data is what we call waves 3 plus 4. And then the whole harmonized data set is all waves 1 through 4. Cumulatively, this is over 900 specimens from about 800 patients and yields a very large data set, about 60,000 clinical annotations, 500,000 drug sensitivity data points, and trillions of DNA and RNA bases that were sequenced. From all of these data, we can do a lot of different things. I won't have time to talk about all of it, And in fact, I'll only highlight a few things, but it's all in that Cancer Cell paper. We can divide the data up into that waves 1+2 versus waves 3+4, and using it as a train test paradigm, and we can show that a lot of those predictions that we made in the first Nature paper hold up very nicely when we look at the waves 3+4 data with a very high level of concordance. We can leverage the entire harmonized data set to extract additional statistical power and reveal new associations that we were not able to see in the original data set. We can use deconvolution techniques to infer maturation states, differentiation states of the AML tumors, and understand how these tumor cell differentiation states are correlating with both genetics and drug response. And I'm going to talk more about this in just a second. Sometimes conditionally with mutations, and we can model clinical outcome. So the RNA deconvolution, for this we turn to a lovely Cell paper published by Peter Van Galen and colleagues in Boston, where they use single-cell sequencing to understand the trajectory, the differentiation trajectory of a number of AML tumors. And from this, they were able to establish gene signatures that could predict or could label tumors across 6 distinct tumor cells across 6 distinct maturation states. And so we used those signatures to deconvolute our RNA-seq data, and we found that there were many correlations of tumor cell state with genetics. What I'm going to show you, though, is the correlations with drug response. And so we found that the majority of drugs that we were testing, and if we organize them into drug families, nearly every drug family showed a very strong correlation of response with tumor cell state. And to orient you, in the middle of the slide is venetoclax, the BCL2 inhibitor. And what we see with venetoclax, and this is consistent with reports published by Dan Pollier and Craig Jordan in Denver, as well as some really lovely work from Carolyn Heckman and her colleagues at FIM, that venetoclax tends to work better against tumors that are of more primitive stem progenitor-like state. That's shown in the 2 red boxes at the top. Where it has liabilities is when those tumors are in a more mature state, a more monocytic or dendritic cell-like state, shown in the blue boxes at the bottom. The polar opposite of venetoclax is the HDAC inhibitor panobinostat. That's on the left side of the slide, where you can see that it has actually good activity against the more mature tumors, and it has liabilities in those more primitive states. And there's a whole cluster of drugs on the left that are sort of panobinostat-like, a whole cluster of drugs on the right side that are more venetoclax-like. And there's a very interesting cluster in the middle that actually does pretty well against both of the extremes of differentiation, but has more liabilities in those intermediate stages of cell maturation state. And I'm going to continue to talk about cell state in just a second. If you've had a good idea, chances are somebody else has had it too. And this is certainly true here, where Andy Zhang and John Dick and colleagues in Toronto, working seemingly right around the same time that we were, did really the same thing. They took both our Beat AML data coupled with TCGA data and data from Guy Sauvageau and the Lucigene cohort in Montreal, and performed a very similar deconvolution of the RNA-seq to arrive at similar predictions of tumor cell state, and correlating with drug response came up with some really similar or almost same conclusions that we did, that there are many drugs whose activity is governed based on the maturation state of the tumor. They also showed some very interesting correlations with clinical outcome and these maturation states. And this leads me to talk about our own modeling of clinical outcome using these data. For this, we used a machine learning tool called Random Forest, which is a decision tree to try to pick out what are the most significant features that split clinical outcome for these patients. Before we used the Random Forest approach, though, we took a look at all of our gene expression profiles. These includes those WGCNA signatures that we had derived from the Nature paper, as well as all of these 6 Van Galen cell state score signatures. And we asked, how do all of these different gene signatures correlate with both categorical and continuous clinical variables, as well as with overall survival? And what you can see in the upper right is that there are 2 of those WGCNA signatures from that original Nature paper that have the strongest correlation with overall survival. And indeed, when we use the Random Forest approach, where we input all of these gene expression signatures, all of the mutations, and many of the clinical features, what we found is that some of the strongest features are already known. Age, P53 status were some of the strongest predictors of clinical outcome. But among those, this WGCNA expression signature, what we call module 3, was one of the most significant predictors of clinical outcome in this cohort. So we were interested in what is it about this module, what is this module that might correlate with clinical outcome. We performed what's called a KME calculation that essentially asks, of the 200 or so genes in this signature, how does each gene correlate with the overall eigengene, the overall gene expression module? We found there was a single gene called pair1 that really drove the whole module, and that's shown on the left. In the graph on the right, you can see pair1 itself versus the whole module, and that it's essentially a linear correlation, and both of which correlate with a more progenitor-like state. And in fact, if we look at pair1 as an individual gene, we actually see just as good of prediction as the whole module for splitting clinical outcome. And in fact, it is about as significant as one of the best prior multi-gene signatures, the LSC17 signature. So this is a single gene which could, in theory, be targeted in contrast to a multi-gene signature. And there's a lot of things that are exciting about considering targeting of pair1. Notably, if we look at the mutational signatures where we see increased pair1 expression, it has many of the worst players in AML, P53, splicing factor mutations, RUNX1, GATA2, MECOM, etc. And it's perhaps not surprising that if you look on the right, then if we look at our secondary AML cases that are transformed from MDS, they have significantly higher expression of pair1 compared to both de novo and therapy-related cases. So I have a couple more slides, but I want to first orient you to how I'm starting to think about all of these data, which is kind of a back to the future, where for many years, as everyone here knows, I'm sure, the French-American-British system was used to categorize maturation states of AML tumors, and that's largely been done away with in the last couple of decades in favor of genetics. But perhaps we should think about returning to some form of FAB, because we can begin to organize drugs into cell states in which they are most effective. Of course, we can then look at extrinsic signals that might be driving cell transitions, which could be targeted, both tumor extrinsic and intrinsic programs. And all of this could be looked at in the context of mutations that could be either driving cell maturation states or could be independent of them. So in the last minute that I have, I want to talk a little bit about some of the drug combination work that we've done. Alongside all the single agent screening, we've tested a panel of drug combinations. One of these has been the approved venetoclax plus azacitidine combination, so we can credential how well does this drug combination ex vivo activity compare to what's known clinically. And it lines up quite well. We can look at mutations and know that IDH, NPM1, for instance, that are more effective, respond better clinically. We also see better responses ex vivo. RAS mutations that are more refractory clinically, we also see more refractory ex vivo. The cell maturation state in this case can be read out with clinical immunophenotype of the tumors, where we can see that the more primitive tumors, CD117 positive, are more sensitive. The more monocytic CD14 tumors are resistant. And we had a small number of cases, 13 cases, that we tested ex vivo and were treated with ven plus azo, and we can see that those that achieved a CR had a much better ex vivo response than those that were refractory. So we can look at all of the different venetoclax-based combinations that we've tested, and we can see that there are some that look about the same as ven plus azacitidine, but there's quite a number that look a lot better. This is looking overall, but I want to drill down into one that looks on the surface not so exciting, ven plus quizartinib. If we look, though, at some of the clinical subsets, such as secondary AML versus de novo, we can see that ven plus quizartinib is the only one that's actually better against the secondary AML cases compared to the de novo. We can also see that it is the only one that is better against ELN adverse versus favorable, and we can see that it is the only one that is better against MLL rearranged AML. So there's utility in looking at how broad a combination might be, but also can drill down to see some exciting subset features of some of these combinations. And finally, I'm over time, so I just want to leave you with, again, the cell state features of these combinations. We can divide them into types, where there's type 1 combinations that look a lot like venetoclax, better against the more primitive. We have type 2 combinations, even though they include venetoclax, are actually better against the more monocytic tumors, and type 3 that are intermediate and look pretty broad. And so again, I'll just conclude with this image, what I think summarizes the data, that as we think about drug combinations, perhaps we can think about pairing drugs that are better against different cell states, or can target transition signals and trap cells into a vulnerable cell state. And with that, I'll stop, and I just want to thank the huge number of people, both outside of OHSU and internal to OHSU, who have enabled this platform to come together. Thank you very much.
Thank you very much. Then our next speaker is going to present via Zoom. Is Dr. Abdel-Wahab from the New York, from the Memorial Sloan Kettering Cancer Center in New York, and is going to speak about therapeutic targeting of RNA splicing factor mutant MDS.
So much, everyone. I greatly apologize that I'm not there in person, and I really want to thank the organizers for the opportunity to participate virtually and for you all for your attention. So my name is Omar Abdel-Wahab, and I'm going to talk about therapeutic targeting of RNA splicing factor MDS. So as many of you are very familiar with, mutations in genes encoding RNA splicing factors are highly recurrent in patients with MDS. In fact, they are currently the most common class of genetic alterations in MDS. And the three most commonly mutated splicing factors in MDS are shown on this slide here. And what you'll immediately appreciate is that in these three genes, the mutations occur almost entirely as heterozygous point mutations at very specific amino acid residues. So for example, in SRSF2, all the mutations occur at the proline 95 residue and affect almost nowhere else in the open reading frame of the gene. Secondly, in diseases such as MDS, where each of these genes individually is mutated in about 15% of patients, it's extremely rare to find patients with hotspot stomatic mutations in these genes, two of these genes simultaneously. So there's this mutually exclusive pattern of these mutations. And these two facts together suggest that these mutations might confer some kind of gain of function or change of function. And what that might mean for their role in the pathogenesis of the disease is very intriguing, and it also highlights them as a potential therapeutic target. So in the last 10 years or so since the discovery of these mutations, what we've learned about functionally their impact is that they are indeed change of function. They alter RNA splicing preferences in a manner distinct from loss of function, and they alter splicing in a sequence-specific manner. And I'll illustrate what I mean by that in a few slides. Excitingly, we're also beginning to uncover that in some cases, these mutations generate aberrant, unannotated mRNA sequences, some of which the proteins that they encode may be required for disease maintenance themselves. So in the heterozygous state, we now know, based on human genetics, that these mutations are highly selected for in myeloid malignancies, such as MDS and AML, and then clonal hematopoiesis, as you heard from Stein. Interestingly, however, we've also learned from cell line models and animal models that when we deviate from this heterozygous state, these mutations are no longer tolerable in cells. And just to illustrate that, these are bone marrow sections from mice with various perturbations in SRSF2. So if we delete one allele of wild-type SRSF2 or express a heterozygous point mutation, it's generally very well tolerated in animals in vivo. However, if we delete the wild-type allele in the setting of the mutation, there's now a complete failure of hematopoiesis, and the mutation is not tolerated for cell survival. Similarly, if you were to co-express two hotspot mutations in splicing factor genes simultaneously in the same cell, a situation which virtually never happens in patients, this is also not tolerated in cells. And just to illustrate this point, we now have animal models for each of the activating mutations of the splicing factor. We can express them individually in the hematopoietic system, and animals are born at normal Mendelian ratios if they express a single splicing factor mutation. However, those animals that have two mutations simultaneously in the hematopoietic system from earliest hematopoietic development die at 100% embryonic lethality due to a failure to develop definitive hematopoiesis. So altogether, how we interpret these data is that while these mutations may be selected for in the heterozygous state for whatever reason, the cells that contain these mutations cannot tolerate further genetic perturbations to the process of RNA splicing. And this concept, which has been seen by multiple groups, has led to this idea that if we could chemically modulate splicing, we could maybe elicit this same synthetic lethal relationship, and this might be a therapeutic strategy for splicing factor mutant MDS. Now, the challenge has been trying to translate this into any kind of clinical effective therapy. there are a number of drugs which are in early phase clinical development. Probably the furthest along is in phase 2 clinical trials currently, showing this blue box here on the right to target splicing in a variety of different ways. And so for the remainder of the time, I wanted to tell you some interesting updates into some of these therapeutic approaches targeting RNA splicing, mainly looking at a really interesting new update on SF3B inhibitors and a little bit about click inhibitors, which I know you'll hear about in much more detail in the talk after mine. So for SF3B inhibitors, there are a variety of natural compounds originally derived from bacteria, and then synthetic analogs have been generated which bind to the mammalian SF3B component of the spliceosome and prevent its interaction with RNA to block splicing. This resulted in development of a orally bioavailable SF3B inhibitor drug called H3B8800, which is still currently in phase 2 clinical trials, but largely didn't have the efficacy that was hoped to see in at least a phase 1 study. But one important thing to know is that H3B8800 and the other molecules shown on this slide here are natural products which are incredibly complicated in their chemical structure. And they have, no doubt, limited PK and PD properties in vivo in human beings, and have limited target inhibition in patients as a result. Although there was some modest degree of splicing modulation in vivo, they are limited in their PK and PD, and that really cannot be overcome based on their chemical properties. However, in the last 2 weeks, in collaboration with Benjamin Kravat's lab at the Scripps Institute, there's now been discovery of an actual small molecule inhibitor of SF3B complex. And this is actually a covalent inhibitor of SF3B1. It's a true small molecule. Its chemical properties, PK and PD, are completely distinct from the drug shown on the prior slide. This is proteomic data on the top right here, where if you pull down the proteins bound to this molecule, the only protein that comes down most selectively is SF3B1 itself. In vitro, this drug perturbs RNA splicing, very similar to H3B8800 and its predecessors, such as pletionolide B here. So our hope is that this molecule, which is now attempting to be translated into a therapeutic and early phase clinical trial, maybe will have very different activity than we've seen with these other prior older SF3B inhibitors based on natural products. Now, of course, I have no idea what a covalent small molecule of SF3B1 will do in terms of safety, toxicity, or efficacy. But the idea here is that this is completely chemically distinct and molecularly distinct in how it inhibits SF3B1 compared to what we've had previously. The other aspect that I'm really excited about is this topic, again, which you'll hear about more after my talk, is inhibiting kinases which are required for proper RNA splicing. And the pathway that's been targeted currently most effectively are these kinases that are dual specificity kinases that phosphorylate tyrosine and serine 3 residues from the click and DIRK family of kinases. So these are two family of kinases that are very related to one another. Their kinase domains are highly similar to one another. There are 4 click kinases and 5 DIRK kinases. And the main substrate that's known about currently for these kinases in cells are RNA splicing factors. So for example, the SR protein family, which includes SRSF2, are heavily phosphorylated by click and DIRK kinases on their serine residues. And each of these is phosphorylated at many different sites within a single protein by these kinases. And so being kinases, there are a number of small molecule kinase inhibitors that target them. Some of them are shown in this slide here. There are at least 5 inhibitors of click and DIRK kinases, which are in clinical development currently. I want to highlight one which has been billed more publicly as an IRAK4 inhibitor, quote unquote, but it also targets click kinases, click1, as potently as it does IRAK4, and this is this CA4948 molecule from Sekeres. This is actually a click inhibitor as much as it is an IRAK4 inhibitor. Others shown here, we worked with BioSplice Therapeutics to develop a click and DIRK inhibitor, which hits clicks and DIRKs equally, but a number of other kinases shown here. And then you'll hear about Cordia's drug, which is highly selective for click and DIRK kinases over any other kinases in the kinome tree here. And there are several other companies that have similar molecules in development, each of which differ a little bit in their selectivity for click and DIRK kinases over other kinases in the kinome. But each of these drugs, when you place them on cells, causes a dose-dependent reduction in phosphorylation of RNA splicing factors. And this can be read out by an antibody that recognizes phosphorylation sites on all SR proteins as shown here. And when these are applied to cells, there's a dose-dependent perturbation of splicing where you have these increases in intron retention due to impaired splicing throughout the genome. And that's shown here on these dots here, where each of these dots represents an intron, which is not being removed in the presence of drug, which, for example, with a drug like venetoclax shown on top, does not perturb splicing as expected. So we eagerly await to see what happens with the clinical development of these drugs. But one last point I'll make is that all the drugs I've talked about to date, while they may hopefully have some kind of synthetic lethality with splicing factor mutant cells, they do target splicing in all cells to some degree. But we know that these mutations, as I mentioned in the outset, are a change of function. So for some time, we've also been trying to address this question shown here, which is that rather than targeting wild-type splicing activity, can we actually target the gain-of-function mutants? And so one way that we've been endeavoring to do this is we've developed recently this platform technology that we're calling synthetic introns, where we identify missplicing events which are uniquely induced by the splicing factor mutation, and we use these to interrupt the protein coding sequence of a gene of interest, such that when this sequence is introduced into cells, cells with the splicing factor mutation will recognize this sequence as an intron and excise it to drive a full-length open reading frame and protein production, whereas cells without this mutation will fail to remove the sequence and not produce the protein of interest. And this actually works remarkably well. We can monitor this by looking at fluorescence protein expression. And so using a sequence that mutant SF3B1 recognizes, and the starting sequence we see here that the mutant SF3B1 cells express far more fluorescent protein than wild-type cells due to the differential recognition of the sequence and removal of the intron. We further optimized this sequence, and now we've generated sequences which are fully efficiently recognized by the mutant cells and not at all by the wild-type cells. So with these sequences in hand, what we've been trying to do is actually use these to deliver selective therapeutics and cellular toxins and suicide genes to eliminate splicing factor mutant cells. And so just to illustrate that, the earliest iteration of this is we've used the suicide gene of HSVTK, or the herpes simplex virus thymidine kinase, which has been used in a number of gene therapy studies previously. This is an enzyme that's normally inert in cells, but in the presence of ganciclovir antiviral medication, HSVTK phosphorylates ganciclovir, and then ganciclovir interrupts DNA synthesis to kill dividing cells. And so the concept here is that HSVTK would only be expressed in the splicing factor mutant cells, and those would only be sensitive to ganciclovir, whereas wild-type cells would be impervious. We've tested this in a number of preclinical models, so in cells with and without an SF3B1 mutation, they're introduced into mice expressing this vector, and then the animals are treated with ganciclovir or control. So those animals engrafted with splicing factor wild-type leukemias, they are not affected by ganciclovir treatment, and they die 1 month after the start of the experiment. Those animals engrafted with SF3B1 mutant leukemias and treated with control die at a similar time frame, but those animals engrafted with SF3B1 mutant leukemias and treated with ganciclovir now actually have complete elimination of the tumors which engrafted in the mice. And this is because these tumors uniquely expressed HSVTK, which was not seen in the wild-type tumors, and so responds to ganciclovir treatment. And so the issue here, last slide is...
Sorry, we have only 1 minute left because we are off time.
Sure, I'll summarize. So mutations in splicing factors are highly recurrent in MDS and confer a change of function, as I mentioned, and they are preferentially sensitive to perturbations in splicing. There are a number of drugs that exist to perturb splicing with distinct modalities, and I highlighted these new covalent SF3B inhibitors. Then you'll hear about the promising data on click inhibitors next in this sort of gene therapy approach. So thanks again for your attention.
Thank you. Thank you very much. Our next speaker is Dr. Yoda from the University of Kyoto in Japan, and he's going to present a novel drug, a novel splicing inhibitor targeting myeloid neoplasms, CTX7112.
Thank you very much. Today, I will talk about our CLK inhibitor, CTX712. Okay, this is my GUI. Dr. Omar already mentioned, several years ago, we identified frequent mutations, splicing factors in myeloid neoplasms, most commonly affected SS3B1, SRSF2, UTF1, and GRSL2. These mutations were mutually exclusive and except for those in GRSF2 affected only one allele. So suggesting that the further perturbations, splicing functions, might be toxic to leukemic cells or cause synthetic lethality in these leukemic cells. So we therefore decide to develop novel compounds that inhibit RNA splicing. So we targeted CLK kinases, which are involved in the phosphorylation of splicing factor SR proteins, such as SRSF2, which is essential for RNA splicing. So finally, we developed a novel CLK inhibitor, CTX712. CTX712 is an orally available small molecule that potentially inhibits the CLK family of kinases at the concentration of 100 nanomolar and is highly selective for CLK kinases out of 468 kinases tested, showing more than 100-fold selectivity over other kinases except DYRK kinases. In Western blot analysis using the PAN phospho-SR antibody, CTX712 was shown to inhibit the phosphorylation of several SR proteins in a dose-dependent manner, which was also demonstrated for SRSF2 using an anti-phospho-SRSF2 antibody. To investigate abnormal splicing induced by CTX712 treatment, we next performed RNA-seq analysis using the MV411 cells before and after CTX712 treatment. We found that CTX712 treatment induced prominent exon skipping in MV411 cells in a dose-dependent manner, suggesting that exon skipping is the predominant alteration in RNA splicing. Sashimi plots showed an example of drug treatment-induced exon skipping. We calculated a percent splicing PSI value. A higher PSI value indicates the frequency of the inclusive isoform in a particular exon-exon junction. So these exons were highly skipped after drug treatment. To summarize the first part of my presentation, CTX712 is a highly selective, orally available CLK inhibitor. CTX712 strongly inhibits the phosphorylation of SR protein. And CTX712 induced a profound splicing abnormality, which were dominated by exon skipping. Next, we investigate the relationship between splicing and drug sensitivity using primary AML cell in ex vivo. First, we investigate effect of CTX712 on leukemia cell growth using the primary AML samples. So in which primary leukemia cells were treated with a different concentration of CTX712 and cell survival was measured to determine IC50. We also performed RNA-seq analysis of these primary AML cells before and after drug treatment. We observed prominent abnormal splicing that was dominated by exon skipping as seen in MV411 cells. To quantify the effect of CTX712 on RNA splicing, so we measured the percent splicing in PSI for all exon-exon junctions and calculate the median difference in PSI, delta PSI, before and after drug treatment. Of interest, clustering analysis of RNA sequencing data revealed that AML samples were clustered into two distinct subtypes according to the splicing changes caused by the drug. The sample clustered in group high showed the larger splicing changes of skipped exons and higher sensitivity to the drug. As shown in the scatter plot, median delta PSI correlates with negatively with IC50, suggesting that the greater the splicing changes after treatment, the greater the growth inhibition. We investigated the relationship between splicing factor mutation and the drug sensitivity using the primary AML MDS cells. CTX712 showed anti-tumor activity not only in splicing factor mutated cell, but also in splicing factor unmutated cells. However, we observed poor response in TP53 mutated cells. The anti-leukemic effect of CTX712 was also tested using the PDX model. So in which mice were inoculated with the PDX cells and then treated orally with CTX712 for 2 weeks. A total of 13 subcutaneous AML MDS PDX model were tested. So we quantified the drug efficacy by evaluating the percentage of tumor growth inhibition, TGI, at day 15. So with higher TGI value indicating the greater sensitivity. CTX712 was able to induce a complete response in 2 models and significant tumor growth inhibition in 12 out of 30 models. The right-hand side shows an example of the PDX carrying the mutated SR receptor compared to mock-treated tumors. So large subcutaneous tumor can be seen in BECO-treated mice, which were ultimately reduced to unmeasurable level after CTX712 treatment. Tumor regression was dose-dependent. Minimum body weight loss was also observed. So these results suggest that CTX712 has an anti-tumor activity at a tolerable dose. We also performed RNA-seq and splicing analysis and investigated the relationship between drug efficacy and the splicing changes. So in the scatter plot, we also observed the positive correlation between the magnitude of the splicing changes induced by CTX712 and suppression of the leukemia growth. So these results are consistent with those obtained in the EXB assay. Conclusion, CTX712 is a novel, orally available, highly selective CLK inhibitor. CTX712 potentially inhibits the phosphorylation of SR proteins, induces profound exon skipping. CTX712 exhibits anti-leukemic activity in vitro and in vivo. The event of the abnormal splicing induced by CTX712 possibly correlated with the efficacy, suggesting that CTX712 induced anti-leukemic activity by inducing abnormal splicing. CTX712 is effective not only in splicing factor mutated cases, but also in splicing unmutated cases, while it is less sensitive in TP53 mutated cases. The identification of reliable biomarker predicting efficacy is needed. Phase 1 trial is ongoing in Japan, so this is data reported in the previous ASH. Out of 8 relapse or refractory AML MDS cases, 4 CR and 1 CRI were observed, and overall response rate was 62.5%. In some patients, long-lasting responses were observed. CTX712 showed preliminary anti-tumor efficacy for both AML and MDS. I would like to thank all contributors. Thank you very much.
Thank you. Thank you very much. Our last speaker is Dr. Aitane Díaz Marquianar from the University of Pamplona in Spain.
Okay, good afternoon, everyone. Okay. First of all, I would like to thank the scientific committee for accepting this abstract as an oral communication. I have nothing to disclose. So, as a brief introduction, myelodysplastic syndromes are characterized by presenting ineffective hematopoiesis in the bone marrow due to an aberrant maturation and differentiation of hematopoietic stem and progenitor cells, HSPCs. The vast majority of the molecular studies that have been performed in this field have mainly focused on describing the mutational status of the disease. And even though in recent years transcriptomic studies have arisen, there are still few transcriptomic studies performed on individual HSPCs. That is why this project aims to offer more detailed study about the molecular pathology of MDS to better understand early myeloid differentiation at the transcriptional level. The biological question we aim to answer is, why is this myeloid differentiation process altered in MDS? We hypothesized that the dysregulation of the activation of several genes or regulons in these HSPCs could be altering the differentiation process in MDS patients. So understanding these mechanisms would allow the identification of new potential therapeutic targets. Therefore, the main objective will be to characterize transcription alterations and gene regulatory networks that could be responsible for, for aberrant hematopoiesis in MDS patients. Regarding the experimental design we have followed, we have obtained bone marrow samples from 12 healthy donors with a median age of 70 years, as well as bone marrow samples from untreated MDS patients that presented multilineage dysplasia, 18 individuals in total. From these bone marrow samples, we FACS-sorted HSPC subpopulations, these 4 cell types. We conducted a bulk RNA-seq, and we performed an exhaustive computational analysis. First of all, we wanted to know what was different between the healthy and MDS HSPCs. So first of all, we conducted a conventional differential expression analysis in the HSPCs in different cell types, comparing MDS versus the healthy condition. When we did an overlap of all these genes, we observed that the vast majority of the differentially expressed genes in MDS were cell type specific. This means that across early hematopoiesis in MDS, different genes are being altered in each differentiation stage. Actually, there were very few genes that were commonly altered in all HSPCs in the disease, and all of them were widely described. So, following the aim of this project, we wanted to uncover the genes that are altered across early hematopoiesis in MDS patients. So for that, we studied 2 different differentiation trajectories. On the one hand, the GMP trajectory that gives rise to monocytes and granulocytes, and on the other hand, the MEP trajectory, which gives rise to megakaryocytes and erythrocytes. So once we wanted to study both differentiation pathways, we developed a computational model that identified genes that showed an aberrant expression pattern in the disease. What does this mean? Well, imagine that you have a certain gene for which we have recorded these expression patterns for the healthy condition in gray and the MDS condition in purple. So at the first glance, you would say, well, this gene is downregulated in MDS. It is differentially expressed. But if you focus on the expression pattern that it follows in both conditions, both are identical. So our model won't identify this kind of genes. Instead, imagine that we have another gene that instead of showing the biologically expected expression pattern for MDS patients, that would be the expression pattern in dotted line in pink, we acquire a different pattern that is no longer identical to the pattern observed in the healthy condition. These kind of genes will be identified by our model. And the fact that these genes are yielding aberrant expression patterns along early hematopoiesis in MDS make them potential contributors of the disease. So next, we quantified the number of genes with altered dynamics for both trajectories. and we clustered them depending on the type of disruption that they were suffering from one stage to another. And as you can observe, those genes belonging to cluster 5 and cluster 8 were the most representative ones. Which are these genes? Genes from cluster 5 are those that show a progressively higher expression than the biologically expected in MDS, whereas those from cluster 8 are genes that show a lower expression than the biologically expected in these patients. Therefore, as these were the most representative clusters, we performed an enrichment analysis in these genes. And we discovered that those genes belonging to cluster 8 that showed a lower expression than the biologically expected in MDS were involved in neutrophil activation and degranulation in the case of the genes identified for the GMB trajectory, whereas those identified for the MEP trajectory were participated in gas transport. So in overall, these results suggest that cluster 8 genes that are detected in both pathways were related to key processes for proper myeloid cell activation and functionality. So next, we wondered if it existed a master factor that could be driving such transcriptional dysregulation in MDS. So for that, we used TRER, a computational method developed by our group that is able to infer regulatory networks that show an altered activity in MDS with respect to the healthy condition. Especially, we focused on the regulons, that are the associations between a certain transcription factors and its associated target genes. TRER identified 175 regulons that showed an altered activity in MDS. Among these regulons, we identified several transcription factors that had been previously associated with hematological malignancies and had been widely described. And in addition, we observed how, doing several survival analyses with the Cancer Genome Atlas samples, we observed that it existed an association of these transcription factors with poorer survival rates in AML. So these findings validate the fact that TRER is able to identify not only regulons that could contribute to the pathogenesis of MDS, but could also serve as potential biomarkers for more aggressive entities like AML. But we wanted to offer novelty to the study, so we focused on SMAD2. This transcription factor was negatively correlated to its target genes, and interestingly, among its target genes appeared genes from cluster 8 that were having a reduced expression than the biologically expected for MDS. And indeed, they were implicated in myeloid cell development. There is very few known about SMAD2. We only know that it is involved in pre-mRNA splicing and is a component of the Spliceosome, but its role in MDS is still unknown. So next, we wanted to uncover which was the role of SMAD2 in MDS. Taking into account that this transcription factor is not upregulated, it's not differentially expressed in MDS, we decided to conduct CRISPR inhibition experiments in HL60 cells, where we infected these cells with control viruses as well as with viruses that expressed guide RNAs against SMAD2. We left these cells in culture for 18 days, and then we assessed different myeloid markers, myeloid differentiation markers, by flow cytometry. Here, for instance, I am showing you the gating strategy for 2 myeloid markers, and we observed how, when we inhibited SMAD2 with 4 independent guide RNAs, we observed that the inhibition promoted myeloid differentiation in these cells. So regarding ongoing work, we know that we need further validation of these findings. We are currently doing additional validations of CRISPR inhibitions, not only in HL60 cells, but also in other AML cell lines. We are also conducting gene silencing experiments in primary CD34 positive cells from MDS patients. And we also want to validate these findings in larger cohorts of MDS patients in order to do potential associations with other clinical variables. So in conclusion, we developed a computational model to identify genes whose expression dynamics are altered across early hematopoiesis in MDS. Most of these genes were not differentially expressed. Ontology studies in cluster 8 genes indicated their implication in myeloid cell activation and functionality. Transcriptional rewiring inference by TRER identified regulons with aberrant activity in MDS that could contribute to the disease phenotype and serve as potential biomarkers. Among identified transcription factors, SMAD2, which was not differentially expressed in MDS, had a key role in myeloid differentiation abrogation in AML cell lines and could represent a potential therapeutic target for MDS patients. In overall, this work offers a new approach in the study of the molecular pathogenesis of MDS and reveals novel transcription alterations and regulons involved in the disease not described to date. I would like to thank all the people that has contributed to this project, all collaborators, the MDS Foundation, funding resources, and patients and donors. Thank you very much.
Thank you very much to all the presenters. Even that I expected to have an amazing answer and questions regarding, of course, the treatment, early treatment in LEN with LEN in Del5Q, we don't have time to realize this answer and questions part of the talk because we have to do some change for the next session. So thank you very much for all the presenters, and we move to the next presentation in a few minutes. The organization says that they are able, that you are able to take some lunch boxes outside the auditorium for the next meeting. If you don't want and you don't need to go, if you don't like, to the exposition area. Thank you very much.